what do you mean by carbohydrate? Before that, we know what is carbohydrate because you all are studying biology. You all studied biology in lower classes also. So we all know what is carbohydrate, right? Yes. See, what do you mean by carbohydrate? See, carbohydrates are optically active polyhydroxyaldehyde. This is the chemistry. Got it? In biology, we are studying, yeah, carbohydrates are there, different types of glucose, sucrose, starch, that's all, finished. But here we are going to discuss what is the chemistry of these carbohydrates. So carbohydrates are optically active polyhydroxyaldehydes or ketones. See, I discussed about this word optical activity. What do you mean by optical activity? If a compound is able to rotate the plane of plane polarized light, plane polarized light, then we can say the compound is optically active, right? You remember this one. This one we studied in aldehydes and ketones. So if a compound is capable of, if the compound is able to rotate the plane of plane polarized light, either in left or right direction, then we can say the compound is optically active. Then this compound can rotate the plane into either right or left, I told you. If it is left direction, we can call the compound as levorotatory. If it is in right direction, it is called a dextrorotatory. Okay, levo and dextro. Okay, then see, so they are optically active polyhydroxy. What do I mean by polyhydroxy? Hydroxy means OH group, right? Polyhydroxy means more than one OH groups, more than one hydroxyl group or more than one OH groups. Then aldehydes or ketones, not only the OH group, the carbohydrates contains OH group, hydroxyl group, aldehyde group and keto group. Or keto group means in it, sometimes it may be aldehyde, sometimes it may be keto group, means CHO group or CO group. Okay, then compounds produced by hydrolysis produce such units. Got it? So carbohydrates means they are optically active, then they contains hydroxyl group, then aldehyde groups or ketone group, depends. Okay, then what is the source? We know that the source are, importantly the source is plants. Okay, then what is the main example for this type of carbohydrates? Cane sugar, glucose, sucrose, starch, all of them are examples for carbohydrates. Got it? Yes. So we know what is polarized optically active compound. We know what is aldehyde group. We know what is keto group, and we know what is hydroxyl group. Okay. Yes. Classification. So we know what is carbohydrate, right? Now we are moving to the classification of carbohydrates. See, carbohydrates can be mainly classified into two: monosaccharides and polysaccharides. Okay. So what do you mean by monosaccharides? See, a monosaccharide is a carbohydrate that cannot be hydrolyzed further to provide a simpler unit of polyhydroxyaldehyde or ketone. See, we know what is monomer and polymer, right? In chemistry, we are studying about what is a monomer. Monomer means, see, polymer means it is made up of different, means many number of single units. And the single unit is called a monomer and the bigger one you are getting is called a polymer. Same thing is here. Monosaccharide means monomer we cannot divide again, right? Same thing is here. Monosaccharide means this one we cannot divide into simpler unit again because that is the basic unit. So it is called a monosaccharide. Okay, there are around 20 monosaccharides are known to exist in nature. For example, glucose is a monosaccharide. Fructose, see, glucose means C6H12O6. This is an example for monosaccharide. Got it? See, monosaccharide means we cannot divide it again. We cannot divide into simpler unit of polyhydroxyaldehyde or ketone. Got it? Yes. Then what do you mean by polysaccharides? It will be opposite. See, polysaccharide means we can convert them, we can divide them into simple units. Okay, so polysaccharides are the carbohydrate that release large number of monosaccharide molecules upon hydrolysis. When you are breaking the polysaccharides, when you are trying to break a polysaccharide molecule, they will convert into, they will produce more than one monosaccharides. That is called a poly. Poly means many. Polysaccharides means they contains many monomers. They contains many monosaccharides. Got it? Then starch, cellulose, glycogen, and gums are examples of polysaccharides. Okay. So monosaccharides means the best example is glucose, fructose, ribose, like that. Polysaccharide means starch, cellulose, glycogen. All of them are example for polysaccharides. Then polysaccharides not have a sweet flavor. Then they are also known as non-sugars. Normally sugars have a sweet taste, right? But polysaccharides, they do not have a sweet taste. Okay, that's why they are called a non-sugars. Polysaccharides are also called a non-sugars. But these monosaccharides are normally, they have a sweet taste. They are normally sugar itself. Okay, then the classification of carbohydrates can be also done reducing and see. I told you carbohydrates means we know what is a carbohydrate, polyhydroxy, aldehyde or ketone. Then the carbohydrates, based on the number of these monomers present, we can classify them into monosaccharides and polysaccharides. Then we can classify them into another one, that is reducing and non-reducing sugar. Then what do you mean by reducing sugar? Any carbohydrates that reduce tolerance reagent and Fehling solution. You remember tolerance test and Fehling test, right? What do you mean by tolerance reagent? What is tolerance reagent? That is ammoniacal silver nitrate solution, right? In the chapter aldehydes and ketones, we discussed about tolerance test and Fehling test. Tolerance reagent means they are actually ammoniacal silver nitrate solution. This Fehling test and the tolerance test we are normally using to distinguish between aldehydes and ketones, right? Yes. So the Fehling test. Fehling solution means it is a equal mixture of Fehlings A and B. I told you, you remember. So Fehlings A means it is copper sulfate. Fehling B means it is sodium potassium tartrate, right? See, the same thing we are using in case of aldehydes and ketones. We can use the substance here also. We can use the reagents here also. Okay, so they will reduce the tolerance and Fehling solution. You remember aldehydes, when treated with the tolerance reagent, they will produce silver mirror. When they are treating with the Fehling solution, they will produce a reddish brown precipitate. But ketones, they do not react with the tolerance reagent, or means they are not providing the precipitates. So reducing sugars are means reducing sugars, they will give the same result with the tolerance and Fehling solution. Okay, yes. Then oligosaccharides. So the carbohydrates, we are classified into mono, poly, then oligosaccharides. What is oligosaccharides? Oligosaccharides are carbohydrates that when hydrolyzed releases 2 to 10 monosaccharide molecules. Important. Very important. They are further classified into disaccharides, trisaccharides, tetra, and so on, based on the number of monosaccharides produced. See, oligosaccharide means when try to do the hydrolysis, they will produce more than one units, means more than one single units. So they can be again classified into di, tri, tetra based on the number of monosaccharides, based on the number of monomers in which they are producing. Okay, so the two monosaccharide units formed by the hydrolysis of disaccharide may be the same or dissimilar. See, suppose an oligosaccharide is there. On hydrolysis, they are producing two monomers. Maybe the two monomers may be same or may be different. Got it? Means they may be same or dissimilar. Understood? Yes. So monosaccharides, means the polysaccharides, monosaccharides and oligosaccharides. Okay. Now see monosaccharides. Based on the number of carbon atoms and the functional group they contain, monosaccharides are again categorized. So monosaccharide means we know they are not hydrolyzing again. They are only single unit, right? Means glucose, it is a monosaccharide. So these monosaccharides can be again classified based on the functional group. I told you in all the carbohydrates, the aldehyde group may be there or keto group. Both of them are not together. Hydroxyl group should be there. Then the other one may be aldehyde or keto. So a monosaccharide is called an aldose when it contains aldehyde group. It will be ketose when it contains keto group. Got it? Yes. So there is the two classification of monosaccharides. First one, monosaccharides can be classified into two. First one is aldose. Second one is ketose. Aldose means the group is aldehyde group. Ketose means keto group, CO group. Okay, then based on the number of carbon atoms. See, if the number of carbon atom is 3, generally we can call it as triose sugar. Tri means 3. Okay, if they hydroxyl group should be there anyway. If they contain aldehyde group, they are called a aldotriose. Aldotriose. If it is keto group, that is called a ketotriose. Then what is tetrose? 4 carbon atoms. That is called, if it is the group is aldehyde group, aldotetrose, ketotetrose. If it is 5 carbon atom, common name is pentose. But the pentose may be aldopentose or ketopentose. Aldopentose means the group is aldehyde group. Ketopentose means the group is keto group. Got it? Then 6 carbon atom. What is the name of 6? Hex, right? So it is commonly hexose sugar. But hexose sugar means they may be aldohexose or ketohexose. Okay, then same. Heptose means aldoheptose or ketoheptose. Got it? This is monosaccharide. So monosaccharides means they are only one unit. We cannot hydrolyze them into many units again. It's a single unit. So they can be, they may contain more than 3, 4, 5, 6 or 7 carbon atom. So we can call it as triose, tetrose, pentose, hexose, heptose. Based on the means aldehyde or ketone, we can call it as aldose sugar or ketose sugar. Okay, that's all. Then see, one important monosaccharide is glucose, right? We know that glucose. Glucose means they have the formula C6H12O6. Okay, glucose occurs naturally both free and mixed forms. It may be free in nature or maybe in mixed form. It can be found in sweet foods and honey. Ripe grapes have a high concentration of glucose, sugar. Okay, understood, right? So glucose means they are naturally they are occurring in, we can see that they may be free state or in mixed form. Okay, many fruits like ripe grapes, then honey, then sweet foods, everything they contain glucose. Okay, next one, preparation of glucose. How can we prepare glucose? Very, very important. Preparation of glucose is again very important. See, first one, glucose can be prepared from sucrose. Sucrose means C12H22O11, sucrose. That is called cane sugar. We know that, right? Cane sugar. When sucrose is cooked in an alcoholic solution with weak hydrochloric acid or sulfuric acid, glucose and fructose are obtained in equal proportions. Got it? This is sucrose, that is C12H22O11. When hydrolysis means we are breaking with the water. In acid medium, that is H+. H+ means the medium is acid medium, weak acid. Hydrochloric acid or sulfuric acid. Then you will get, see, the formula is same, right? See, the thing is, this is not two glucose molecule. Glucose molecule and fructose molecule have the same formula, C6H12O6, that we know, right? See, then what is the difference between them? There is a structural difference. Between glucose and fructose, there is a structural difference. Okay, but anyway, the formula is same, C6H12O6, C6H12O6. So don't misunderstand like, see, the sucrose is producing 2 molecules of glucose. No, 1 molecule is glucose. This may be glucose and this is also, this is not glucose, this is fructose. Don't think like we are getting 2 molecules of glucose. Okay, yes. Then, next one, from sucrose it's clear, right? Next one is from starch. Okay, glucose is commercially, means industrially, prepared by hydrolysing starch with a dilute acid at 393 Kelvin under pressure. Okay, see, this is starch. Okay, then what you are doing? Same hydrolysis, you will get n number of, because it is a polysaccharide, right? Starch is a polysaccharide. So, the starch on hydrolysis, we are getting many number of glucose. There is no fructose, only glucose. Okay, whatever the number of molecules here, you will get same number of glucose. But in from sucrose, you are getting mixture of glucose and fructose. But from starch, you are getting only glucose. Got it? Yes. Next one is structure of glucose. What is the structure? So we know glucose contains hydroxyl group is there, or aldehyde or keto group should be there in carbohydrate, right? Then see, glucose is dextrose. It is an aldohexose. So aldohexose means it's clear that they contains aldehyde group, right? Glucose, often known as dextrose. It's an aldohexose. Aldohexose means they contains 6 carbon atom. They contains aldehyde group. So 2 informations we got it, right? Yes. It's a monomer of several bigger carbohydrate, including starch and cellulose. See, starch, it is a polymer, means it is a polysaccharide. Starch is made up of glucose, means glucose is a monomer of starch, or cellulose, means another one is cellulose. Okay, it's most likely the most abundant organic compound on the planet. We know that. The general structure of glucose, because as a result of photosynthesis, the plants, they are producing glucose. So it is the most abundant organic compound, right? Yes. So this is the structure of glucose. See, here you can see aldehyde group. Aldehyde group means it will be CHO. Then here carbon, then hydrogen will be there, OH will be there. These unit will be 4. Then the last one is CH2 OH. Okay, that's why the formula is C6H12O6. Got it? It generates N-hexane after prolonged heating with hydrogen iodide, indicating that all 6 carbon atoms are connected in a straight chain. See, all these carbon atoms are in a straight chain, right? Yes. Got it? Yes. Then glucose interacts with the hydroxylamine to generate an oxime. Oxime, very, very important, which is then combined with a molecule of hydrogen cyanide to form cyanohydrin. This reaction shows that glucose contains carbonyl group. See, this is glucose, right? Yes. When it is reacting with the, see, interacts with the hydroxylamine. They are reacting with the hydroxylamine and producing oxime. Okay, this is called a oxime. Oxime. This oxime, what we have to do on reaction with the hydrogen cyanide. Oxime on reaction with the hydrogen cyanide, they will produce a cyanohydrin. This is cyanohydrin. Okay, this reaction shows that glucose contains carbonyl group. That's why this compound is getting. So this is a structural elucidation of this glucose. Okay, then when glucose react, so this reaction is clear, right? Glucose react with hydroxylamine produces oxime. Oxime on reaction with hydrogen cyanide produces cyanohydrin. As I told you, there is no mechanism, only the reaction. Okay, when glucose react with the light oxidizing agents such as bromine water, it is converted to 6 carbon of carboxylic acid. That is called a gluconic acid, not glutonic acid, gluconic acid. This suggests that aldehyde carbonyl group is present. Got it? We got two proof for the existence of carbonyl group, right? So this is glucose. When you are adding bromine water, we are getting gluconic acid. Means this aldehyde group is converted into COOH. Means the aldehyde group is oxidized into acid group. Aldehyde group is oxidized into acid group. That's why they are producing gluconic acid. Okay. Yes. Next one. The existence of 5OH group is confirmed by the acetylation of glucose with the acetic anhydride. How? This is glucose, right? This glucose is reacting with the acetic anhydride. Then what will happen? All these OH group will convert into, see, anhydride COCH3 groups. It means there is 5OH group. Got it? Here see 4OCOCH3, here 1. Total 5 groups are producing. Means the alcoholic group, OH groups are converting into this anhydride, this 4, this C, this 4. Means there is 5 OH group. Okay. When oxidized with nitric acid, glucose and gluconic acid both give saccharic acid, or we can call it as saccaric acid. Saccaric acid. Okay, it is a dicarboxylic acid. Why saccaric acid is called a dicarboxylic acid? Because they contains 2 COOH groups. This implies the presence of main alcoholic group in glucose, because the alcoholic group in they will normally convert into acid group by oxidation, right? So it means they contains alcoholic group. Got it? That's why they are converting into gluconic acid. It is a saccaric acid. It is a dicarboxylic acid. Okay, both the glucose will produce saccaric acid, gluconic acid will also produce saccaric acid. So normally in the glucose, if the one COH group is there, other one is CH2OH, that is gluconic acid. If the CH2OH group is also converting into acid group, that is called a saccaric acid. Okay? Yes. Then Fischer arrangement, again important. Fischer arrangement is there where gluconic acid and the saccaric acid are represented. See what is happening here. This is the first structure. This is glucose, right? Yes. So this glucose is converting into gluconic acid. Gluconic acid is converting into saccaric acid. Okay. Then D plus glucose is the right term for glucose because they are optically active, I told you. Okay, the letter D before the name of glucose symbolizes the configuration. I told you because the dextrorotatory, levorotatory means the positive represents the dextrorotatory. I told you the carbohydrates are optically active compounds. They can rotate the plane of plane polarized light. So what is the difference? See, this is plus glyceraldehyde. This is minus glyceraldehyde. See the configuration. Hydrogen here, OH here. In this one, minus 1, OH is here, hydrogen is there. That is the difference. Got it? Yes. Then, plus glyceraldehyde isomers have a D configuration and L configuration is for the negative isomer. As I told you, see, due to the optical activity. Okay? That's all. Any doubt in this one? Okay. Then we are moving to the cyclic structure of glucose. What is the cyclic structure? Okay. See, despite the presence of aldehyde group, glucose phase shifts reagent. See, you are doing the practicals, right? So you are using the shifts reagent. That the violet coloration, that one. Okay. See, they doesn't create the hydrogen sulfate addition product with the sodium bisulfate. This one you are doing in the practicals, right? The fact that glucose pentacetate doesn't react with hydroxylamide as that the absence of free CHO group means the CHO group present in the glucose is not free. Means they are connected to somewhere. There is a cyclic structure. See, as we show before, it will be like this because the CHO group will be at the top, then the next one it will be CH2OH. Means this group is free, right? But actually, this group is not free. Means that is connected to somewhere. That is the cyclic structure. Okay, glucose has discovered to exist 2 distinct crystalline form, alpha and beta form. Alpha form of glucose is obtained by crystallization from concentrated glucose solution at 303 Kelvin. Important. See, the glucose can exist in 2 forms, that is alpha form and beta form. How this alpha form is getting, how beta form? See, the alpha form is obtained by crystallization from concentrated glucose solution at 303 Kelvin. Crystallization, take the concentrated solution. Concentrated solution means we are not adding too much water. So from the concentrated glucose solution, we are doing crystallization. Then the glucose we are getting should be the alpha form of glucose. Why these alpha and beta forms are different? It is mainly different in their melting point. See, alpha form have 413, 419 and the beta form have 423. Okay, then how the beta form is getting? It is obtained by the crystallization from heated and saturated aqueous solution at 371 Kelvin. Understood the method of preparation? Alpha form and beta forms are the two important forms of glucose. We are getting these two forms in different condition by different method of preparation is there. Okay, so alpha form is obtained by the crystallization of strong means concentrated glucose solution, and the beta form is by the crystallization from heated and saturated aqueous solution. Saturated means we cannot dissolve more glucose. We are preparing the saturated solution. From that saturated solution, we have to do the crystallization. And the first one is the highly concentrated glucose solution. At 303, this is 371. The temperature is important. When the temperature is changing during the method of preparation, the product will change. Got it? Yes. It was found that glucose forms 6-membered ring because we know because it is C6H12O6, right? So it is a 6-membered ring, and that of the OH at the carbon 5 contributes to the formation of the ring. Then how the ring structure is forming? This explains the lack of CHO group as well as the presence of glucose in two forms. See, understood? See, this OH group is utilized for making the ring structure like this. Okay, so you have to know the cyclic structure also. That is very important. Okay, then the only difference between the two cyclic hemiacetal forms of glucose is the position of hydroxyl group at C1 known as anomeric carbon. Okay, difference between two cyclic hemiacetal. You remember acetal and hemiacetal, right? So two hemiacetal forms of glucose means where they are changing at C1. C1 means the first carbon. Okay, such isomers, namely alpha form and beta forms, are referred to as anomers. So anomeric carbon means it is C1. So they are called anomers. The isomers we are getting is called anomers. Okay, so the 6-numbered cyclic structure of glucose is referred to as pyranose structure, and it is parallel with the pyran. You know pyran, right? This is pyran. So from this pyran, you can get, using pyran, you can get the structure for glucose. If you are getting the structure using the structure of pyran, that is called a pyranose structure. Pyranose structure. Okay. Pyran is a cyclic chemical molecule having a ring containing 1 oxygen atom and 5 carbon atom. We know that because it is a 6-carbon compound. The cyclic structure of glucose is better illustrated by the Haber structure, which is seen below. See this one. Okay. Yes. So this is called a pyranose structure. Then did you hear about the furanose structure? Furan. Okay. Pyranose is here. Furanose is different. Okay. Yes. Then fructose. So glucose is clear, right? Now we are moving to fructose. We know both glucose and fructose have same structure, same formula C6H12O6. Then what I told you, there is a difference between the structure. There is a structural difference, right? Then what is the difference? In case of glucose, you can see aldehyde group. But in structure of fructose, you can see keto group. See. So it is a ketohexose. Okay, so it is produced by the hydrolysis of the disaccharide sucrose. We know that because when we are doing the hydrolysis of sucrose, we are getting 1 molecule of glucose and 1 molecule of fructose. Then I told you there is a structural difference. Okay, so it is a monosaccharide found naturally in fruits, honey, is the same thing in naturally available materials. Naturally, we are getting it from naturally. Okay, then structure. So chemical formula is same and contains ketonic functional group at carbon 2. See, the second carbon is keto group. But in glucose, we are not seeing the keto group. It is aldehyde group. Got it? Then 6 carbon in a straight chain. It is a levorotatory chemical of that DC. It is written as D negative. Same thing due to the optical activity. Okay? Yes. Now, it also exists in two cyclic forms which are formed by adding OH group at the C5, means the fifth carbon. So I told you furanose structure, you remember? I told you the glucose is existing in pyranose form. That is the ring structure. Then in case of fructose, it is furan. That is furanose structure. Okay, the resulting ring has 5 members and is known as furanose after the chemical furan. This is furan. Furan is a cyclic molecule with 5 carbon atoms and 1 oxygen atom. However, structures are used to illustrate the cyclic structure of 2 anomers of fructose. These two. Okay, then D plus. What is the difference here? See, here nothing, no difference here. But see the first carbon. This is CH2OH, OH here. This is CH2OH, OH down. This is the difference. Okay. Yes, into disaccharides. Okay, disaccharides. See, disaccharides we know. What is disaccharides? They contains two monomers, right? If you're reducing sugar of monosaccharides such as reducing groups of monosaccharides like aldehydes or ketonic group, we know that because they contain aldehyde or keto group are found bound in disaccharides. They are not reducing sugar such as sucrose, as I told you before, right? Yes. Then sugars that have these functional group free such as maltose and lactose are known as reducing sugar. So I told you during the classification, I told you we can classify them into reducing and non-reducing sugar also. So maltose and lactose are called a reducing sugars. Okay. Then example is sucrose. It is a common disaccharide that when hydrolyzed yield glucose and fructose, that I told you because there is a structural difference. I told you, right? So sucrose is an example for disaccharides. It means on hydrolysis they will convert into two units, two monomers, glucose and fructose. So glycosidic linkage between C1 of the D-glucose and C2 of the beta-D-glucose holds these two monosaccharides together. Sucrose is a non-reducing sugar, as I told you. Okay, and the bond between them is glycosidic linkage. See, one monomer is there here. This is not the actual structure. I am making this to understand you. Okay, then see, to know this concept, I am drawing this one. This is not the correct structure. Okay, so these two units are, so disaccharide means they contains two separate units. So these two separate units are bounded together by glycosidic bonds. Glycosidic bonds. That is the thing you can see here. Glycosidic linkage. Glycosidic bond or glycose. So in disaccharides, the two units, right? They are linked together by glycosidic linkage. Okay. Next one is maltose. Maltose is another type of disaccharide. So sucrose is clear, right? So next type of the disaccharide is maltose. It is made up of two alpha D glucose units. See, sucrose is made up of two, means one glucose and one fructose, right? Here it is made up of two glucose units itself. Here C1 of the glucose unit 1 is joined to C4 of another glucose. See, here this is the first carbon. So the connection between two molecules is the first carbon of this one and the fourth carbon of the next one. That is called a C1 C4 linkage. Same linkage, glycosidic linkage. Okay. Yes. So maltose is a disaccharide. It is made up of two molecules of glucose. Sucrose is another disaccharide made up of one glucose and one fructose. Anyway, the formula is same, but the structural difference is there between glucose and fructose. Got it? Next one is lactose. Next disaccharide is lactose. It is widely known as milk sugar. We know that one. Lactose sugar we know, right? Yeah. So because it is found in milk, it consists of alpha means beta D galactose and beta D glucose. The bond is formed by C1 of galactose. See, here is also C1 C4 linkage, the first carbon of galactose and fourth carbon of glucose. But the monomers are Galactose and glucose. Again different. See, these two are different. Got it? This is different. Different sugars. One is galactose, other one is glucose. So don't be confused about the monomers of different disaccharides. Okay. Means the sucrose means 2 glucose. Okay. Lactose means 1 galactose and 1 glucose. Okay. Then maltose means 2 glucose. Don't be confused. Next one is polysaccharides. So what do you mean by polysaccharides? As I told you, polysaccharides means, see, so between the individual units there is a linkage called a glycosidic linkage, I told you. Between the individual units in carbohydrates, the linkage is called a glycosidic linkage. So what is a polysaccharide? We know many monomers are there, right? So glycosidic bonds hold many monosaccharide units together to form polysaccharides. Okay, in nature, these are carbohydrates that are most frequently found. We know that. Okay, then one best example for a polysaccharide is starch. Okay, plants' primary storage carbohydrate is starch. We know that because carbohydrates are storing in the plants in the form of starch. Okay, then the cereals, tubers, roots, and some vegetables have high starch content. We know the potatoes. Okay, so means these root vegetables, they contains majority of them contains starch amount. In alpha glucose polymer composed of 2 components, amylopectin and amylose. Again, amylose is water soluble component that accounts for around 15-20% of starch. Then one more is there, amylopectose. Okay, amylose and amylopectin. Amylose is a long unbranched chain composed of 200 to 1000 glucose units. See, oligosaccharides means it is between 1 to 10 only. Poly means many monomers are there. See, 200 to 1000 number of monomers are there. But the linkage is C1, C4, glycosidic linkage. The linkage is same in every carbohydrates between the monomers. It is glycosidic linkage. Got it? Yes. Next one, cellulose. Next type of this one, polysaccharide is cellulose. We know that. What is cellulose, right? It is found only in plants and is the most prevalent organic material. The plant wood, it is the important component of the cell wall of the plants. That we know because we are studying in biology, right? Yes. Cellulose is straight chain polysaccharide made up of entirely beta-D glucose units linked together by glycosidic connection between C1, C4 linkage. Again, the linkage is C1, C4 glycosidic linkage. This is an example, cellulose. Okay. Next one is glycogen. Glycogen. The animal body stores carbohydrates as glycogen. Important. The plant body is storing carbohydrates as what? Glucose. Animal body is storing as glycogen. It's also known as animal starch because its structure is similar to amylopectin but more strongly branched. Because there should be a branch between the individual molecules, right? Yes. It is present in brain, muscles and liver. Enzymes change glycogen into glucose when the body requires it. Yeast and fungi also contains glycogen. Okay. So cellulose, starch, glycogen. Is it clear? Carbohydrates are a major portion of our food which gives us energy. That we know. In plants, they are stored as starch. In animals, they are stored as glycogen. Bacteria and plant cell walls are made up of cellulose. This one also we know, right? We make a furniture out of the cellulose in form of wood, and because wood means they contains this higher amount of carbohydrates. Okay. They supply raw materials to a variety of key business, including textiles, paper. Because paper, it is making from wood pulp. This is carbohydrate. Okay. So carbohydrates have very much importance in different fields.